My name is Professor Kyle Lampe. I'm an assistant professor in the Department of Chemical Engineering, and I'm very happy to have with us today Professor Michael King. Professor King is a native Virginian, and he is both an alumnus of our department, as well as serving as a current professor of practice. So he retired from Merck, a national or international pharmaceutical company, after 32 years working there, and almost immediately began a second career as a professor here at UVA. He currently teaches a course on vaccine engineering in our department. And so he has a lot of expertise to bring to the role of chemical engineers in vaccine development, particularly with the aspect of COVID. I believe his very last trip that he took cross country in February of 2020 was to visit the Bill and Melinda Gates Foundation for a meeting about this coronavirus thing that people had been starting to suspect might be a problem. He is a consultant for the Bill and Melinda Gates Foundation, as well as a consultant for the World Health Organization about their COVID response. And I will let him take it away from there. Thanks.
Thank you, Kyle. Let me know if you can hear me. Is that all right?
Yep, we hear you great.
All right. So Kyle's kind of, I'm just going to go here. Somehow not advancing here.
Yeah, you can use your arrow button, perhaps.
That's not working either.
Oh, interesting.
There we go.
Okay.
The other way. Okay. So Kyle covered a lot of this, but anyway, I was born in Bristol, and then as you can see, I was undergraduate at Virginia Tech. I got my master's here at UVA. Then I joined Merck and actually went after 3 years, came back and got my PhD at University of Delaware. And my career at Merck was mainly between research and manufacturing divisions, but the last year I worked directly for the CEO. And after a 32-year career at Merck, I came to UVA first as the Halsey professor, and then currently a professor of practice. And I have been consulting for about 6 years with the Bill and Melinda Gates Foundation. And my interests are travel. I've been all 7 continents, and this is me at Ernest Shackleton's grave down in Robertson in South Georgia Island near Antarctica. This is Walking Jabu in Botswana, and this is Tiger Nest Monastery in Bhutan. So, and then interests are photography and astronomy in addition to the other parts. So you've seen this graph before. This is from Johns Hopkins and shows that the case. When I started teaching my course last year in January 6th, there were 59 cases and no deaths. And now this is just a few days ago, almost 3 million deaths globally and 134 million cases. So a truly amazing pandemic. But there have been pandemics before, and I put this slide up. So you see the 2.7 million with COVID-19 here in the front. And you can see some more recent ones: swine flu, SARS, which is a respiratory, Ebola, so only 11,000, MERS is 850. HIV/AIDS has actually killed 25 to 35 million, okay, which is 1981 to present. There's still no vaccine after 36 years. Then you see Asian flu, the Spanish flu, which everybody remembers was back in 1918, 1919, that killed 40 to 50 million. And then going all the way back to, you look at the bubonic plague back in 1351, it was 200 million, and the population was a lot less then. So there have been a history of pandemics that happen across the world. In COVID, the response to COVID, chemical engineers played a lot of different roles. Vaccines, which I'll talk about today and give you a little background. Also therapeutics, such as monoclonal antibodies, or therapeutics, which are drugs, as well as monoclonal antibodies. Then making the materials to make these products. So media, resins, filters, sterile use, single-use bags, personal protective equipment, process design to facilities to make these manufacturing operations. So there was a huge number of thousands of chemical engineers that have been involved in the response to COVID-19. The challenges is unprecedented global emergency, speed, efficacy, safety, capacity. We're looking at 8 billion doses across multiple platforms by the end of this year. They're now talking about 8 to 10. And at that's a huge number of doses, as you can see, produced each day. This is 24 hours a day, 7 days per week. And so this 10 billion doses is on top of the 4 to 5 of normal vaccines, such as measles, mumps, rubella, polio, yellow fever, etc. So that's a tripling of global manufacturing in 1 year. So obviously that stressed the system as far as being able to actually execute. And so that's been one of the big challenges that we're still grappling with. Around the world, there's a whole team of chemical engineers working, trying to debottleneck and to address this issue. This came from the Washington Post. This is the speed. The goal originally was to do it in 12 to 18 months. We ended up doing it's like 10 months. And if you look historically in vaccine development, chickenpox, which was also at Merck, took 34 years. The shortest one was mumps. This is the Gerald Lynn strain of mumps. That was the shortest at 4 years. And HPV is Gardasil, human papillomavirus, which I hope you guys have taken. That was took 15 years. I was part of that for about a decade. And so the average time was around 10.7 years. So if it normally takes 10 years, how do you squeeze out 9 years without taking shortcuts? So that's probably the question everybody asks. That's the reason to go for this before we start going through the rest. I say it's a convergence of many things. First, pre-investment and response. I'll talk about that. Plug and play vaccine technology, which is new, that wasn't done before. First of all, lots of disease. That may sound strange, but when you have lots of disease, you can do clinical trials much faster because you get a signal between your control and your placebo, so you can pick up the clinical signal very quickly. Lots of parallel clinical trials and activities. Lots of regulatory flexibility, streamlined approval process. This is the first time you've heard the currently approved Moderna and Pfizer vaccines and the Janssen are all being applied with the emergency use application. This is the first time that's been done for vaccines. Normally you'd have to go through some approval process. So huge change there. Knowledge sharing, this was been one of the big heartwarming things of this whole last year is just the global collaboration, because I would be on calls with hundreds of people around the world, and the sharing of information, best practices and whatever has been outstanding. And quite frankly, it's just leadership, commitment, and hard work. I've never seen so many dedicated people. Amazing. We get up at 2 a.m. in the morning, we get up late at night because there's time zones all over the world. And so you have to accommodate the different time zones. The other thing that I'll point out here, there's a lot of other pre-investments that I mentioned, the speed of the response. This was very important. Back in 2009, I'll show you, the US government under CDC trained early detection in China, Korea, and Thailand. This is called the PREDICT program. The National Institute of Health, the National Institute of Allergy and Infectious Diseases, National Institute of Allergy and Infectious Diseases. The Bill and Melinda Gates formed a vaccine group to look at unmet vaccine needs around the world. And then the CEPI, which was 2007. This is Coalition for Epidemic Preparedness. So this was started about several years ago. The Global Alliance for Vaccine Immunization. The US had Warp Speed. And then these new the vaccine technology development platforms, which have been in development for about 20 years. And then rapid genetic sequencing came in the late 2000s, which was very important. And then computer-aided design of the antigens were all critical. So all these things just came together at the right time for a rapid response. The other thing was we saw it coming. Historically, I mentioned the Spanish flu that happened back in 1918. And more recently, the severe acute respiratory syndrome, or SARS, that happened in 2002, 2008. The swine flu, which is pandemic 2009, 2010. Middle East respiratory syndrome, that's MERS. That was also a coronavirus. That was in 2012. And then Ebola was not a coronavirus, but that was an outbreak in Africa in 2014, 2016. So disease X, there will be another disease X, and we have to be prepared for that. Not to scare you, but this is kind of from a world at risk. This is from the World Health Organization, looking at the emerging diseases. And you can see on the pointer over here, of course, SARS came from China. Also, there was others as far as the MERS. was more in the Middle East. But where's SARS? Here's SARS coming in. This also came very close by to where the coronavirus came from. And then all over the world, you have what are called zoonotic diseases. And these were viruses emerged from, moved from an animal host to a human host. And so that when that happens, they used to be contained locally. But things have changed. There's now a global aviation network. And so within 24 hours, you can pretty much, a virus can get almost anywhere around the world. So it's pretty, the good news is you can get anywhere in the world in 24 hours. The bad news is a virus can get anywhere in the world in 24 hours. I mentioned the pre-authorization in 2009, this $200 million project led by Dennis Carroll. And what they did is they went around and trained epidemiology in China, Thailand, South Korea, in early detection of zoonotic diseases. And so this is a very successful program that trained people in the world to do early detection of coronaviruses from bats and other animals. And so that set the time, the capability for early detection up where the viruses normally emerge. Now, not to scare you to death, I'll just go ahead and do these. But this is the coronavirus here. This is the family of the coronaviruses. And so the COVID-19 is a similar genomic sequence to other coronaviruses such as SARS, which you see up here, and MERS, which you see down here. So a lot of its molecular structure and its spike protein was understood and known. So the training in Asia with PREDICT allowed rapid identification of the virus and determine its genetic sequence. So it didn't take long. These were bat-based variants. These are the ones that transferred to humans. So this sequence was pretty fast to do this. Compare that to HIV/AIDS, which it took like 4 years to discover the retrovirus that caused it. So this happened, as you'll see on the next slide, in 7 days, not 4 years. So late December 2019, the Wuhan Municipal Health reported a case cluster of pneumonia. It was isolated in a week, January 7th. That's incredible speed. Thailand, also trained by the CDC outside of China, confirmed the first case outside of China. Then the sequence with the genetic rapid sequencing, China publicly shared the genetic sequence, January 12th. That's just a week or so later than when it was detected, which is pretty outstanding. So when that was shared globally, shared on a global database, Barney Graham at the NIH started working on designing the antigen design for the coronavirus's vaccines. And that's what the basis for Moderna and the Pfizer BioNTech, and actually some of the Oxford Novavax and Janssen antigen design came really out of Barney Graham, who owns the patent at the NIH for the coronavirus. And by March 11th, it was characterized as a pandemic. So vaccines, what we want a vaccine to do is to maximum realism with minimum danger. You don't want side effects, but we want the vaccine to provide neutralizing antibodies to prevent the spread of the infectious disease. We wanted to induce memory helper T cells and B cells, provide long-term protection, so you want a long duration. And you want to make memory killer T cells if possible, and this can control the infection by destroying the infected cells. And you want to do this quickly at high capacity, high quality, and low cost, because there's 7.7 billion people on Earth, and we need to protect all of them, because no one is safe until everybody's safe. Very quickly, if you look at RNA viruses, they have an envelope spike that are all trimers that sticks out of it. So this is the membrane of the virus, and this is called the spike protein that sticks out of it. It's coronavirus, HIV, RSV, measles, etc. And again, I'm not going to go, this is not an immunology course, but this is showing again the coronavirus, and this is a spike protein on the outside of the capsule. And before people came on, I was talking about the name coronavirus comes when you do a transmission electron microscope that looks like a crown. And so the crown is for crown. That's the name of the virus. So this is the envelope of the virus, and then this is the spike sticking out. And on the end of the spike is this receptor binding domain. And this is what attaches to the human ACE2 receptor and infects humans. So this is the mechanism of infection. And this is just some of the candidates that you can do either just this part, or you can do the full spike. This is what it looks like. This is a stabilized prefusion spike, which is the antigen design for the Moderna, the Pfizer BioNTech, and some of the others. And these are just some of the other subunits you can do of that. You've all had your vaccines, I hope. And so there's examples as far as there's different types of vaccines. There are live attenuated vaccines such as measles, mumps, rubella. There are inactivated vaccines like the salk polio vaccine, subunit vaccines like for hepatitis B or the human papillomavirus or Gardasil, viral vectors such as Ebola, and now we have a RNA vaccine, and it's a new technology. So all of these were used to respond to COVID-19. When you look at it, there were over 337 development programs globally. And if this was like in, I think this is March 14th, there were 12 approved globally, there's 3 in the US. But you can see all the different types of technologies that are under development. And so all huge, most of them preclinical, with 12 of them only in the approval here. As far as the manufacturing experience, there's a lot of experience. These are like developed like in the 50s and 60s, and a lot of known about that. Inactivated later, subunit, and then viral vector and this. So the manufacturing experience, a lot more here, but a lot less over here. But another thing is that these live attenuated, that's biological safety level 3. And so containment requirements, so you want to contain it because you're growing up a large amount of infectious virus. And so this requires a large degree of containment. And there's not a lot of facilities to do this. And so this had a lot of if you had special containment, there wasn't a lot here. So a lot of different processes. So one of the things that happened with coronavirus and why it got approved so quickly are these plug and play vaccine platforms. Instead of developing a process just for that vaccine, there was a platform developed. Janssen, which is part of Johnson & Johnson, had developed a vaccine platform using an adeno26 as a carrier vector for the antigen. And they grew it up in per-C6 cells. They used that for Ebola and then adapted it for COVID-19. So they switched out the antigen from Ebola and put in the COVID-19. And similar for the Oxford program, they had a chimp adeno vector, and they replaced, that was used for actually for Ebola, and they replaced it with the COVID-19. Also, for about 20 years, people have been working on messenger RNA vaccines. And multiple companies, Moderna, Pfizer, BioNTech, CureVac, developing these, and which the genetic code to make the antigens inserted into your body, and your body actually makes the antigens, and I'll go through that example. So these are two different things. So these are platforms. They're not just developed for any one disease. They can be used for almost anything, depending on what antigen you plug in. Key to the messenger RNA working was the delivery system, and this just happened in the last 2 or 3 years that made it possible. And it's called a liponanoparticle. And so the messenger RNA is put on the inside of this little fat bubble, if you want to call it, which has these different lipids that form like a 60 to 80 nanometer particle. And this is the delivery formulation for the antigen, to get the antigen to the cells. That's the basis for the Pfizer BioNTech, and this is from the New York Times. They've had some wonderful graphics on how these vaccines work. And I'm not going to spend a lot of time, but this is the gene for the spike protein in the coronavirus. So they clone that gene out, and they actually make messenger RNA. I won't go through that whole process, but then they make the messenger RNA for that spike protein, and they put it in one of those liponanoparticles I just talked about. So this is the messenger RNA for the spike protein. It's the code. It's really the code to make the spike protein. So that's what's injected into your body. So this vaccine particle comes in and attaches to one of your cells, and then the messenger RNA comes in. Once inside the cell, the ribosome, it translates it into the spike protein. So your own body, think of this as a thumb drive with the code to make the protein. And you're inserting that thumb drive into your cell, which then uses that information to make that protein. So there's no virus involved. This is essentially telling your own cells how to make the exact spike protein that's on the virus. So those are made, they actually assemble, there's 3 trimers that come together to make a spike. And that can be either protrude and extend on the outside, or it can be displayed depending on the type of cell it is outside the cell. When the cell dies, it will actually also release these fragments of this protein and peptides. They're picked up by something called an antigen presenting cell, and that's near the point of injection. And after they pick up this foreign protein, they start migrating. They migrate to the lymph nodes in your body. They travel. And at the lymph node, they turn on then what's called your helper T cell. And this is part of your adaptive immune system that initiates the immune response. The T cell then goes on to activate a B cell. B cells are the cells that make antibodies. They start secreting antibodies. And then the antibodies then kind of attach and neutralize the virus so it can no longer infect and stops it from infecting. T cells can also turn on what's called a activated killer T cell or cytotoxic T cell. So the antigen presenting cell can turn those on, and they actually go and kill cells that are infected with the coronavirus. So you're taking out the coronavirus both by antibodies, neutralizing antibodies, as well as by having cytotoxic T cells that take out the infected cells. And this is the study from the Pfizer vaccine efficacy of 95%, which is unheard of. People from the World Health Organization, we were hoping for 50%. And so everybody was really happy when it turned out to be 95. So this is the first dose of the Pfizer BioNTech. And you can see just after a little over a week and a half, you start seeing the people it start flattening out. For those that had the placebo, the saline placebo, they kept getting the disease. And so you can see the separation between those who got the vaccine were protected, these were not. And so clinically, it worked incredibly well. So why did it finish first? Okay, this is again the spike protein design of the antigen. Rapid computer enhanced design of the stabilized prefusion spike protein. This was done actually at NIH by Barney Graham, and then it was tweaked a little bit by Moderna and Pfizer. It was only 70 days from the sequence to injecting in someone's arm. That is unheard of. Normally that would take years. So that was really fast. And the reason that was allowed was because they already had clinical studies underway showing that the plug and play platform was safe to do this. Okay. Then 370 days, less than a year after the sequence was released. Remember, the sequence was a week after it was identified. Okay. You had efficacy of a vaccine. That is incredible. Okay. That's less than a year. First readout of high efficacy, and this was made possible by high rate of infection, because very quickly by that graph I showed you, it became clear it was unethical to continue that clinical trial because the people that got the placebo were at risk of catching the disease. And once you knew that it worked, it's unethical not to stop the trial and instead to give the people that had the placebo the vaccine to protect them. And these are the first human messenger RNA vaccines. And likely they certainly will not be the last, because this is probably going to be a fast response platform of choice for the future. And there are other vaccines coming. The Janssen's out now. There's one Novavax that there's a protein subunit looks really good that should be out very shortly. And there's a ton of other second generation vaccines coming. And the other thing was just doing a lot of things in parallel at risk instead of doing things sequentially. The company started actually manufacturing vaccine before they knew it would work. So hundreds of millions of dollars were at risk if it didn't work, but you couldn't wait to see if it worked and then start up a manufacturing plant. So that was done at risk. So it's a significant overlap of sequence of the trials. And instead of submitting a regulatory license, it was a rolling regulatory submission, as we just saw an emergency use authorization versus going through the final approval process. So this is what this looks like. Up front, I told you about the 10 years, which is up here. So this is what happened for COVID-19. Everything was compressed and overlapped. So you had a huge overcompression of doing things at risk. So you didn't just do a phase 1, then do a phase 2, then do a phase 3. You went phase 1 directly into phase 2, directly into phase 3, and then you had phased approval while you were still continuing your phase 3 data. And so this was obviously much faster. And then manufacturing just was set up where you didn't wait to the end. You actually did it before you had your phase 3 data. So this has never been done before. So this is, again, it's another. So the whole point of this slide is to say that the experience with COVID-19 will likely change vaccine development forever. It'll probably never go back to the old way. So if there's a bright silver lining in this whole episode, is that we're going to have much more rapid response to future pandemics. And I would just like to say that knowledge sharing, global communication was, there is an issue with nationalism, and it's still a challenge as far as what can be exported in and out of what country. These organizations have really paid off as far as this global network. And I will tell you, working on a lot of with the other chemical engineers, chemical engineering is a global language. And when you talk to people in the UK or Japan or China or India, we're all speaking chemical engineering and the things that have to be solved, the problems that have to be solved to make things possible. And it's really heartwarming to see people work so well together. So this network, of global network of organizations. The knowledge sharing that I mentioned was unprecedented. Clinical protocols were published. They never were published that fast before. And things in journals are published real times. And this common problems were leveraged instead of solving them for each program. This organization, which I'm part of, called COVAX, is under the World Health Organization. Really we realized there wasn't enough glass vials, so we did multi-dose images to conserve the limited glass supply, simplified labeling, barcoding, how to do the technical transfers between production facilities, and all these different things. We have another workshop coming up next week between the manufacturing SWAT team and the clinical SWAT team to address some issues with the variants, on how we're going to do variants and introduce them into the supply chain. And so I'll just put all this up. So it wasn't one thing that made it possible within a year. It was the pre-investment that I mentioned, the plug and play technology, especially the messenger RNA, parallel activities, manufacturing at risk, with a high rate of infection, faster clinical readout, knowledge sharing and communication was unlike anything that's happened before with a pandemic, and just a lot of hard work of people around the world. But you got to vaccinate people, so you got to worry about the getting vaccines into people's arms. So I'm a big fan of Gary Larson and his cartoons. So this is the vaccination man prowling in the neighborhoods. Point here is vaccines don't save lives, vaccinations do. So after you make the vaccines, you've got to get it into people's arms. And as you know, that's a struggle we're going through now in the US and around the world. The US is actually now ahead of most of the world. And getting to 75% of 7.7 billion people is a huge challenge. And I don't think any one product has ever been thought of so globally. I don't think any type of product has ever been thought of as a product for the whole global population. Getting the resources to get it to low and middle income countries. But right now, the vaccines are going to the high income countries and it's not getting to the low and middle income countries. And therefore, variants are going to start to emerge if you don't take care of that. So I may hear no one's safe until everybody's safe. And then learning from this so that the next disease X, we're ready for it. So that's happening. There's a lot happening right now on the mutations. Second generation vaccines are underway. There are actually new Moderna, Pfizer, and several programs have already started clinical studies against the South African strain that came up. So there's a lot of work still to be done here. So after that wind up, that's how it was done in 30 minutes, how it was done, the vaccine a year. So what do you need to develop a vaccine? You need virologists, immunology, protein, analytical, clinical, regulatory affairs, and chemical engineering, okay? And chemical engineers play a surprisingly big role because manufacturing is very difficult for vaccines. And I won't go through all the details, but normally you have to go from cells and plasma, cell line, drug substance, such as this. And if you look at this is for the human papillomavirus like particle. And this is very complicated. If you look at Zocor, this is like a cholesterol drug. Its molecular weight is around 200 Daltons. The HPV vaccine is 20,000 kilodaltons. So the complexity of making this and making it consistently so that it works the same is much more difficult than making a chemical compound. And so this is an IgG therapeutic monoclonal antibody, and that's 150,000 kilodaltons. So this 150 versus 20,000, you can tell the complexity of vaccines are tremendous. So this is chemical engineering subjects applied to vaccine production. Chemical reaction kinetics, which is what chemical engineers do best. Okay, that's for bioreactors and chemical reactors as well as stability prediction, actually predicting how well the vaccine will be stable. Fluid dynamics and mixing. Mixing is a critical issue to make the lipo nanoparticles. It was a key factor in making the lipo nanoparticles. If the impinging jet mixers that chemical engineers developed had not been completed, we would not have the lipo nanoparticles. We would not have the Moderna, and chemical engineers did that. So I want you to understand that. Transport phenomena, mass transport, aeration, nutrients, heat transport. If you have a lot of cells, like if you look at the universe, Milky Way is 100 billion stars, 12,000 liter bioreactor has 100 trillion cells. So it's magnitude. So you have to, if you grow a lot of things, you have to remove the metabolic heat, cool stability. Professor Geis in the previous one talked about heat transport. It's important to remove the heat from that to maintain it. Then he also talked about separation, unit operations, filtration, centrifugation, chromatography. All the messenger RNAs all have a chromatography step after you've made the messenger RNA to purify out fragments. Extraction, precipitation, crystallization, analytical methods, designing plans, control systems, project management, and ProbStat, probability and statistics, comes in very big in designing clinical trials that you're going to get the right clinical readout. So if you take what chemical engineers teach, all of this applies to vaccine production. So it's actually kind of an essential skill. I showed this slide before, and I just want to show this is where chemical engineers mainly work. It's in the manufacturing, developing the process, small scale clinical trial material, the large scale manufacturing and manufacturing scale up. But you have to do it working in collaboration with your clinical partners, your regulatory partners, and it's not like something you do by yourself. It's a collaborative effort. And so chemical engineers, you have to work with other people. So what I wanted to show very quickly are just some of the people and examples of chemical engineers and what they did in vaccine development and manufacturing. Product development and design, Andrew Biederman, he's now at MIT, and they're developing a new vaccine. Nancy Nidell is at Novavax, that's a subunit protein. And I'll talk about the drug substance and drug product process development. So product development, process development, project management. I'm talking about Dave Robertson and some of the stuff I do. And then manufacturing, Nick Sayer, UVA, Paul Mensah. He also is going to have a talk at 1 o'clock as well, and Hannah Cole Merck. So these are just a bunch of people. I just want to talk about some of the jobs that they did briefly in the COVID vaccine. This is Drew Biederman. He graduated from UVA with honors about 3 years ago. And I think he was in Kyle, he was in your lab, correct? No. It was in Jeff's lab, or?
Yeah.
So anyway, Drew, so he's now at MIT working in Chris Love's lab, and he's working on a new receptor binding domain vaccine, which is going to be a second generation that's under development right now. So he graduated undergraduate chemical engineering. He's now getting his PhD at MIT, and he's working with Chris Love in developing a new lower cost, high capacity vaccine for the world. And so he's a pretty exceptional person. And I just happened to notice his name on one of the slides that I was on a late night meeting with Chris Love. And I said, is that the same Drew Biederman that went to UVA? And he said, yes, and he's doing terrific. He's one of the leaders in the lab. And so he's doing extremely well. This is Nancy Nidell. Knew her when she worked at Merck over at Elkton. And this is the process from Novavax. They actually use insect cells, moth cells, along with baculovirus to produce their S-spike protein, which they then combine. It forms this is an example for Ebola, but it's very similar for the COVID-19. It forms a nanoparticle around 40 nanometers. And so Nancy's working on the scale up of the production of their vaccine. And so I'll be on several calls, and I've seen Nancy see her name pop up, and so we're coordinating together. So she's up in Rockville, Maryland area. So she's working on process development and scaling up manufacturing of the Novavax vaccine. My immediate boss at the Gates Foundation is David Robertson. I knew him also at Merck. David's deputy director at the Bill and Melinda Gates Foundation. His undergraduate was from Berkeley, his PhD's from MIT, and he coordinates between all the vaccine functional areas at the foundation. And he's also the key person between the Coalition for Epidemic Preparedness Innovation, CEPI, and the Global Alliance for Vaccine Immunization, Gavi, World Health Organization, the World Bank, all these type of people. So he's a high-level connector. He provides a lot of technical analysis. One of the first things, well, I'll talk about when I did it, but the manufacturing landscape to support vaccines. Assume that the chemical and manufacturing and chemistry regulatory issues are chemistry, manufacturing, control, regulatory issues are addressed. He's also on the COVAX technical review group and the manufacturing SWAT team that are solving problems across programs, and also identifying and mitigating supply issues. So I talked to David Thursday, and we went over some different things that are really a challenge right now as far as consumables that are affecting supply. This is Nick Sayer, who's a fourth year UVA Chemie. This last summer, Professor Geis was talking about internships. He was an intern at Lonza, which is a contract manufacturing organization for Moderna. And he was up at Portsmouth, New Hampshire, and he got to do actually the large scale up of the messenger RNA at Portsmouth, New Hampshire at the Lonza facility. And he had a great experience there. So this is Nick. Paul Mensah, he's a senior vice president at bioprocess research at Pfizer at St. Louis. And this was on St. Louis Public Radio. They had an article on him, and he's in charge of manufacturing and process development. So he had a lot to do, especially with the production of the messenger RNA. So he made the plasmids, of which has the DNA to make the RNA. I won't go through all the details here. They had a big role in that. And this is another article that came from UVA today. And Paul's going to have a talk at 1 o'clock, and I hope he can do that, because he's going to talk not just about, he didn't have time to get one released on vaccines, but he'll talk about generally what chemical engineers do in a biopharma career. And so Paul's an exceptional person, and he got his master's and PhD at UVA, and he's doing extremely well. Hannah Knoll graduated, I think it was last year, or year before, and she's at Merck down in Durham, North Carolina, and she's now working, they're going to be making the Johnson & Johnson Janssen drug substance. So they'll be making the viral vaccine down in Durham. And so she's part of that. So she just graduated, so she's part of the COVID response. And then there's little old me, okay? And so I just do help identify risk and then mitigate it, connect expertise. So we need people that know formulation expertise. David Volkin at Kansas University and others. I provide a lot of chemistry, manufacturing, control support to partners of the foundation. One of the first jobs I had was actually a survey of global drug substance and drug capacity in the world. And so we hired a consulting firm. And so jointly, we surveyed all the different companies to find out who had manufacturing capacity and where could we use it, and then we matched up developers with capability. So we're essentially saying, okay, they have the capability here, you can do that. You need to go over here for that. So it was really matchmaking to get the people so we could make stuff. Several committees on the World Health Organization's COVAX, which is the vaccine response. The Research Development Manufacturing Investment Committee, we made about $2 billion in different programs. And then the Open Technical Review, which is issues across programs. And I'm also on the Gavi Independent Product Group, which evaluate all the final COVID vaccines before they're approved to be purchased by Gavi and eventually distributed through UNICEF. So in summary, chemical engineers are essential contributors. Designing effective product. So what I mentioned that Drew Biederman's doing and others, designing manufacturing processes consistently through safe and effective vaccines at both quantity and low cost. That's what Nancy and others are doing. Building startup manufacturing facilities like Nick was doing, tech transfers, identifying potential issues and mitigation strategies, what I and Dave do, and then leadership, cooperating in an intense global, multicultural, multi-technical team effort. And I must say, this is the most fun, because you really have a lot of people that are really putting aside their own interest and working very hard together to solve problems. And to me, that's just very heartwarming to be in those meetings. You come out and just realize how incredible it is to have so many people really focused on doing something good. So with that, I'll open it up for questions. And I guess you can either, there's just a few people on the call, so you can just ask me directly, or you can put it in the chat to Kyle or Gino. I'm not sure how it works.
Yeah, thank you so much, Mike. Yeah, so I think Professor King did a great job showing some of the people. Hopefully you all can identify yourself as one of those people someday in the future who could make such an impactful contribution to this kind of thing, whether it be a future disease or some other aspect of pharmaceutical industry. The first question I see here, Professor King, what are some steps that chemical engineering student can take during college if they want to work in pharmaceuticals or vaccine development?
Well, first, I would just say the core curriculum. Okay, focus on that because the core curriculum can apply to any of the types of work that you saw Professor Geis show. So you have to understand the basics of mass transport, kinetics, reaction, or whatever, crystallization. Then I would supplement that with choosing your electives as carefully. And so as was mentioned, there's a bioconcentration. So you could take courses in bioseparations. You can take additional courses in biochemical engineering. I have the course in bioproduct and bioprocess engineering. Professor Geary has a course in crystallization, which is important in the pharmaceutical industry. So there's different courses that people can take if you want to have a concentration. I would then try to encourage you, if you're interested in the industry, is actually, you don't know what you don't know. And so try it out. And so have a summer internship and see what you like to do. And you may find, because I tell you, during my career, I thought at first I want to work in the lab the rest of my life, and I got in charge of a technical group, and I enjoyed doing that and developing, recruiting people. And you don't really understand your strengths and your weaknesses till you try different things. So having different internships is a good way to do that as well. So I would think do well in your core courses, take select your electives well, and try to have an internship, and then do undergraduate research with someone that's doing part of this as well. So there's all opportunities to broaden your perspective. And I would also encourage, and I haven't talked to the other faculty about this, to take courses outside of the engineering school, if you can, to really get different perspectives, because you see things from different angles. Because I remember one of the courses I took when I was in UVA, my master's degree, I took medical microbiology in the med school. And actually that turned out to be I learned a lot from that. So I just, you sit down with your academic advisor and kind of say, okay, what are the things you can do where you can explore different opportunities?
Yeah, and just to reiterate what Professor King said, your coursework, the core chemical engineering coursework is one type of preparation. The electives are another type of preparation. But then the experience that you can gain, right? So the undergraduate research, which a very large percentage of our undergraduates participate in research, both in our department, in the School of Medicine, in chemistry, throughout the rest of the university, as well as the internships. And you can kind of mix and match internships and research experience. So you can do both of those if you're not sure what kind of direction you might want to go after you finish your degree. Again, feel free to drop other questions in there. I was gonna throw one at you, Mike. In terms of the manufacturing processes involved with the different types of vaccines, can you maybe mention just a little bit more about what differences there might be in those processes and the processing or the scale up, and what independent challenges those might throw at folks?
Yeah, I think the historical, if you take like the live or attenuated virus vaccines, there you have to grow up. First of all, you have to do the master seeds to prepare the viruses. So you'd have to, like for attenuated, you'd have to develop a virus that's been modified so it still will provoke an immune response but won't give you the disease. So that takes long. Then you have to prepare the master cell bank for that. Then you have to grow that up in another cell line. So you have to prepare a master cell bank for that. And then when you grow up the cells, then you infect the cells, and then you have to harvest and purify it. That has to be heavily contained. And so those take a long time, and that takes, as I mentioned, a lot of containment equipment, and that's more difficult. So they have unique challenges with live virus or attenuated or inactivated viruses. For subunit proteins, you can do larger scale in insect cells and yeast or whatever, and then you purify the protein out of that. And then there's generally a formulation and making it into, sometimes there's virus-like particles. You have to go through a folding, refolding. And so those are a little, they don't require that same degree of containment, okay? And that's really more applied recombinant technology and those type of subunits. And then finally, the messenger RNA is actually they clone out the DNA you want. You grow that up in E. coli and plasmids. You purify that the DNA out. You linearize it, and that's the template to make your messenger RNA, which is done in a cell-free synthesis, and then purified by chromatography and then stabilized. So they're each got really unique challenges, but the thing about the advantage messenger RNA is that you can change the antigen in a weekend, whereas to change a live viral and others takes months. So the first vaccines to address the variants are going to come from the messenger RNA. So they have certain advantages over the others.
Great, thanks. Another question from the chat, and I think you're uniquely qualified, having worked in vaccine development for 40 years now. Are there other engineering disciplines that are involved in vaccine development? I'm guessing, the answer is yes, but can you maybe give a hint about why and how?
Yes, the chemical engineers play a big role, especially in the chemistry, manufacturing, control. But obviously, you have to build facilities. So there's civil engineers, there's process design, there's process controls, which take electronic, electrical engineers, and there's a certain amount of I guess a little bit of civil, mechanical. Mechanical is a very big issue. Mechanical, because it may seem simple, but to what's called fill finish, to put the final vaccine into vials, is not a trivial task. You have to do it under aseptic conditions in a very controlled environment, and these are high-speed mechanical lines to fill that, and you can't have glass in there. You can't, broken glass, and you got to make sure the vials are taken care of. So there's a lot of mechanical engineers, control engineers, and you work with a lot of different types of engineering. On the process to make it, though, is more working with, I would say, virologists, biologists, biochemists, and then all of that works with clinical people.
Yeah, and I would throw out, perhaps in addition to that, biological or biomedical engineers, perhaps early in the process in terms of, and maybe less so with the mRNA lipid particle versions compared to the ones that are made in cells, where there's you have to engineer the cells themselves to create something, which would be more analogous to the earlier versions of vaccine production than the current mRNA.
And I could probably add in materials engineering as well, because you remind me the formulation development is an important part in actually understanding because there's people that actually, David Vulcan, who's really great at that. He's at Kansas University. He's a chemical engineer by training, but they do a lot of things as far as formulation, denaturation, analytical characterization, predictability. So there's a lot of skills that go into that. So drug delivery, BME is involved with that, material science, so a lot of different things.
Yeah, someone's got to make the lipids to make the lipid nanoparticles too, right?
Yep.
Another question from the chat. Can you give some examples of chemical engineering companies or jobs in Virginia in this area?
Yes, UVA East, which is, I guess, in the valley. It's called Merck. At Elkton, Virginia, there's a large facility, and they've hired a lot of UVA chemical engineers. I actually was manager of tech service there for a period of my career, and so that's actually where HPV vaccine is made. They also have been making some other vaccines in that area. There's also in Northern Virginia, outside of DC, there's GSK. So there are vaccine companies or pharmaceutical companies in the area. The majority, though, are in New Jersey, I'll be honest, or out at the West Coast. And so and again, it's a global issue, so you'll find the opportunities are everywhere, but there are a few local companies.
Yeah, and there's a couple additional ones that have facilities in the Washington, D.C. area. You mentioned GSK, but I know there's some additional bioseparations and
well, Novavax is just outside of that area as well. So there's a growing biocapability, mainly centered around Rockville, Maryland, which is near where the FDA is. So.
Yeah, and then there's also, I guess we didn't really think about this, but there's a number of governmental agencies that are involved with this as well that are based in the regional area. So certainly the NIH has a significant internal program, as Professor King mentioned. They're the ones that first took the sequence and made the spike protein in their internal capacity, and then also, the FDA is based in the Washington, D.C. area, and NIST, National Institute of Science and Standards and Technology, which does a lot of verification of materials. Can you highlight other chemical engineering jobs that are maybe not specific to the biopharma industry as well?
Any not related to biopharma?
Correct, based in Virginia, yeah.
Oh, yeah, there's a lot. I mean, as Professor Guy said earlier, there's a lot of, if you look like Ashland Chemicals in Richmond, there's several breweries like Coors, Amherst, and what's it? Used to be Coors Brewing. It was bought up by Anheuser-Busch.
InBev.
And InBev now over, and that's in the Valley. But there's different beverage companies, food companies, chemical companies, pulp and paper. There's other different things in the area that chemical engineers can be employed. And I'm sure I'm missing a lot of of course, all the consulting in the banks.
Dow,
Northern Virginia.
Right.
Faulkner, right?
Klockner Penoplast in Gordonville, yes. They do a lot of actually high film for pharmaceutical packaging, and they're high end. So,
yeah. And then the energy industry as well.
Yep.
So there's a number of chemical engineers that work for like Dominion Power, for instance. That's certainly part of it, but there's a lot that we're not coming to mind right at the moment. I think from our particular department, our undergraduate bachelor degree folks, we have on the order of about 40% to 50% that work in the biotech sector of chemical engineering, which is perhaps a little bit higher than the typical department. But we still have lots of people that work in the materials and manufacturing and energy industries, including renewable energy.
Yeah.
So that exhausts our questions from the chat for right now, but I want to thank Dr. King again for giving us an idea of some of the impactful things that can happen as well with a chemical engineering degree, as well as some of the individuals that come from a variety of different backgrounds and a variety of different institutions, but have all contributed in the global response to getting COVID-19 under control. So thank you all, and have a great afternoon.